I suppose the other speakers have said it's hard to see you. It really is with this light right here. It's great to be here, and I'll tell you about the work in our lab that's been going on over the last couple of decades. And we're focused on bioinorganic chemistry, or metallobiochemistry. And this is the study of proteins that have metal ions, or enzymes that have metal ions in them that are needed for catalysis or for structure. But it's also the study of how bacteria or microbes get the metals that they need to grow, and also how other cells maintain their metal ion homeostasis. So we all do bioinorganic chemistry with every breath that we take. And I'll illustrate that in a second. But here, for a typical 70-kilogram human person, let's see if I get this right. Oops. There's about 5 grams of iron, 1.8 grams of zinc, and about 0.25 grams of copper relative to the bulk elements in a person. And in order to see, to illustrate this, take a huge breath. And the oxygen that you've just breathed in is coordinating and binding to the iron and hemoglobin. Don't exhale yet. And because what's happening is that oxygen is being transported to your brain and to muscles, and that's where respiration is going on. You need the enzyme cytochrome oxidase. And cytochrome oxidase has many copper atoms as well as iron heme centers that then reduce the dioxygen down to water, and they're oxidizing glucose back to CO2. So if you ran this reaction backwards, that's photosynthesis that you may be aware of also. But you still can't exhale because you need another metalloenzyme. You need the zinc carbonic anhydrase in order to maintain the equilibrium, the equilibration between CO2 and bicarbonate. And so when you shake a soda can and it fizzes and bubbles and you think that's CO2 being released and building up pressure, you think that equilibration is fast. It's not fast enough for us. And that's why we need an enzyme then that carries that out. Now, we have a lot of iron, zinc, and copper. There's a lot on terrestrial environments. There's a lot of these metal ions also. But the ocean, the transition metal composition is completely different. So the next time you go swimming, think about the fact that the most abundant metal ion in the ocean is molybdenum. Vanadium is the second most abundant metal ion. And by contrast, iron is really, really dilute, not like with us. You know the ocean is salty, so it's half molar in chloride, millimolar in bromide, micromolar in iodide. And also, it's about micromolar in hydrogen peroxide during daylight hours, and this is as a result of photochemical events. So organisms evolve against the chemical constraints of their environment. So what kinds of new metalloenzymes might be present in marine organisms? And that was the question that I asked when I was starting off as an assistant professor. So it's not surprising that a vanadium bromoperoxidase enzyme would be found in marine organisms. It's found in every class of marine seaweeds and in many marine bacteria. So this enzyme uses vanadium for catalysis, and I'll show you how in a minute. And it's using bromide and peroxide to halogenate organic compounds. That's the RH. And so we've looked at the biosynthesis of many brominated, halogenated natural products in marine seaweeds. But in terms of sheer tonnage of brominated or chlorinated compounds produced, it's the volatile halogenated hydrocarbons that are produced in huge amounts by blooms of temperate marine algae. And they correlate with the decrease of the Arctic ozone layer during these blooms. Not the Antarctic ozone layer, because the residence time isn't long enough in the atmosphere, but in terms of the Arctic it is. Okay, so this is a really simple enzyme. You could imagine that there's the vanadium. It's sitting at the bottom of a deep valley, and it's just vanadate coordinated to the protein by one side chain amino acid residue, histidine. The way this enzyme functions then is that the peroxide has to attach to the vanadium. It's bound side on, and in the bound peroxo state, it tunes up the potential so it can oxidize bromide, and it gives us hypobromite. So this is the bromine analog of hypochlorite. So some of these organisms are actually making bleach also during the turnover of this enzyme. And that's exactly what can do electrophilic halogenation reactions of organic substrates. Okay, so why does an organism want to make these halogenated natural products? What good is it for them? In one sense, many of these compounds were known to have chemical defense functions for these organisms. So certain organisms, like a fish trying to eat a particular alga, may not like the taste of what some of these brominated compounds are. So they're ichthyotoxic compounds. But also, if you think about any time you place a surface in water, it becomes biofouled, it's slimy. And that's the result of microorganisms growing on the surface and creating the slime. So how does seaweed keep all these microorganisms from growing on it and fouling their surface? Because they must maintain a pristine surface to do the chemistry to grow. So if you think about it, in 1 ml of seawater, a tiny amount of seawater, there are 10 to the 7th viruses. There are 1 million bacterial cells and 1,000 fungal cells. That's a lot of microorganisms to colonize surfaces that are in the ocean. And so bacteria then, when they're trying to colonize a surface and make a biofilm, they can't do it one bacterial cell at a time. They have to know that there's a cohort of themselves that are out there, and this is called quorum sensing. How would you know that your neighbor is next to you? You can see that, but a bacterium isn't working that way. And so instead, these bacteria shoot out small molecules called homoserine lactones, is one type of quorum sensing molecule. And if it then embeds in the receptor on the surface of another bacterium, the bacterium goes, aha, my cohort is there, let's do something together, let's make a biofilm. So a very interesting area of research actually is how do you disrupt and interrupt quorum sensing so that a bacterial cohort could be growing but not know that their offspring or brothers and sisters are growing next to them. And that's what we were interested in, particularly in whether or not some of these brominated compounds are disrupting quorum sensing. So it had been known that this is a seaweed, Delacia pulchra from Antarctica, although it actually is found in Australia and New Zealand also. And it makes these small molecules, the brominated furanones. And they actually interact at the same receptors on the surface of bacteria that the homoserine lactones. And they are known to disrupt quorum sensing. But what's the role of this vanadium enzyme in making these compounds, and how else could this vanadium enzyme be helping the seaweed to limit biofouling of its surface? So we needed to know where this enzyme is. So if you just take a piece of this alga and put it in a pool of phenol red. So some of you may have carried out titration experiments with an indicator dye phenol red. It appears yellowish at around pH 6 or 7. And then if we add to this whole piece of alga and the phenol red hydrogen peroxide and bromide, we can see the blue color developing in 1 minute and certainly by 3 minutes. And that's the bromination of the compound that produces this indicator dye bromophenol blue. So we know that on the surface, the enzyme is present and active. So that means the question is, what about the molecules that the bacteria is sending out? Could the alga or the enzyme in the alga brominate these homoserine lactones, and particularly the 3-oxo version, because at the 2-carbon position, that's a perfect site for electrophilic halogenation. And the answer is yes, and it brominates either once or twice, and we can certainly find these compounds by mass and isolate them. And it turns out they also disrupt quorum sensing in the bacteria. So I have to tell you about what this quorum sensing experiment is. This is an engineered strain of a bacterium called Agrobacterium tumefaciens. And it's engineered to release an enzyme called beta-galactosidase that acts on a dye called XGAL under quorum sensing conditions. And this XGAL then gets hydrolyzed by this enzyme to form a blue-green compound that's a lot like indigo, the dye that makes blue jeans blue. And in the presence of the brominated compound, we can see the bacterium is growing, but it's not turned the blue color, the release of this dye from XGAL. So then we turn to, well, how might the enzyme be involved in the biosynthesis of these brominated brominated furanones? And we don't know what the original, the substrate is to make these. But we said, what could be the simplest substrate that might give us a brominated furanone? And that was for pentaenoic acid. And we can brominate here, and it can cyclize, and it makes this very simple bromofuranone with just one bromine atom. And that also disrupts quorum sensing. We can see here under quorum sensing conditions, we see the blue-green dye being released, whereas in the presence of the bromofuranone, the bacterium's growing, but it doesn't know that it's under a quorum of itself, and it's not releasing the enzyme. So one way that algae then protect themselves is they're able to disrupt quorum sensing in microbes trying to colonize their surface. And I've shown you one example with this vanadium enzyme that both makes a compound that prevents the biofouling, but also can attack the molecules, certain molecules that bacteria produce. Okay, so that's the story of an abundant transition metal that's in the ocean. What about the story of iron in the ocean? All bacteria have to have iron to grow, with the possible exception of a couple of species. And so this is a false color image of iron, an average over 10 to 12 years of iron concentrations in the ocean. And the deep blue colors are the very lowest levels of iron, roughly picomolar iron. But bacteria typically need about micromolar, 10 to the minus 6 or 10 to the minus 7th molar iron to grow. How do bacteria get iron? Well, if they're growing in a host like us, they can often get the iron out of hemes or out of the iron transport proteins, transferrin or lactoferrin. But if they're growing out in the environment where there's not much iron, they synthesize small molecules called siderophores, Greek for iron-liking. And there are these groups called catechols that bind quite tightly to fold up and bind around the iron in an octahedral fashion. And then they're recognized by specific receptor and brought back in. So we wanted to know, what are the siderophores of marine bacteria? And when we started this, there weren't really any that were known. And so, here's a map then of where we got water from or where we collected it ourselves. And you can see it's regions of low iron, not the absolute lowest levels of iron down in the southern oceans, although we have looked at some down here. And what we do is we take that water and we streak it on a petri dish which has a blue iron dye. And when iron is removed from the dye, it turns an orange-pink color. So these brown spots are the colonies of bacteria growing, and those that have a halo around them have pulled iron out of this dye. And so these are good candidates to say, we're going to pick these up, we're going to grow them up, we're going to find out what the compounds are and what the siderophores are. So that's what we do. We grow them up in 4-liter flasks, and then we sample over time, and we add a little bit of our growth medium to a liquid blue iron dye, the blue cast solution, and when it turns pink, we know we have our compound that's present in solution. And then we harvest and run a lot of columns and run a lot of HPLC, and we isolate the compounds. And the slide I'm about to put up next was years of work, okay? But it feels funny to put it up and just not attribute this to all the students whose really hard work went into determining these structures. So it's not simple, like the one I showed you with enterobactin. So these are peptides. They have one of a series of fatty acids. Well, that sounds pretty good, because maybe the fatty acid can anchor in the membrane, and the head group can hang out to get the iron, and then the bacterium doesn't have to worry about diffusing away in the ocean. And what's been really quite interesting is that a group at Woods Hole Oceanographic Institution now has isolated some of the molecules that we've found in growth directly from the ocean. That's a hard experiment to do because they're present at such small concentrations, but they look at mass and the fractionation pattern of the mass. Okay, so one class then of the compounds we were finding were these suites of fatty acid-tailed siderophores, but another class were photoreactive when they were bound to iron. And that's this group circled in blue. It's an alpha hydroxy carboxylic acid, and when that binds to iron, I looked at this and thought, oh, this is a lot like molecules that photochemists use to count photons. I bet this is going to be photoreactive if we shine light on it. And sure enough, so here's this band in the ultraviolet. Here's the UV visible spectrum of this iron siderophore. We put it out in sunlight, or we shine specific wavelengths of light, UV light, and we get the red spectrum. And what's happened to the molecules, we lost that amino acid and we lost all the fatty acids from there, and now we've produced another compound. So this is photoreactivity of these iron siderophore compounds. And a postdoc in my group at the time, Catherine Barbeau, who's now a faculty member at UC San Diego, showed when she was in my lab that actually the process of this photochemistry makes the iron more available to other microorganisms at large in the ocean. So there seems to be some synergistic relationship there. Okay, so instead then of just getting water and plating it and not knowing what you get, we've now approached things in a much more targeted way through genome mining. So this has been truly fascinating. There are just like thousands and thousands of microbial genomes that have been fully sequenced now. And we can go in and look for what kind of siderophore might we want to study, what might be interesting. And so one that was interesting to us is produced by Vibrio harveyi. Vibrio harveyi is the organism that many people use to study quorum sensing because its readout for quorum sensing is bioluminescence. So when it gets to a sufficient cell density, it glows this beautiful blue color. And it has what looked like everything to make enterobactin, but it didn't make enterobactin. Instead, it had a gene that would encode an enzyme that put a fatty acid tail on it, like our other siderophores, and it makes these anti-enterobactin siderophores. So this core, what's called this serine macrolactone core, is really quite remarkable in siderophores. And so we wanted really very much to target other types of siderophores that were based on this core. And so we found a series that inserted an amino acid between the catechol, so this is the group again that binds the iron, and the serine macrolactone core, and these are what are called the cationic amine siderophores. So I didn't put a plus charge there. There should be another proton and a plus charge there. And that would be the amino acid lysine, but not the typical L-lysine that we would have in our systems, but D, the opposite enantiomer, D-arginine, L-ornithine. And I want you to hold this thought. There's catechols next to amines in these siderophores. And we're going to turn now to the third and last story, which is what other kinds of molecules in marine organisms have this catechol and cation right next to each other, and how do these organisms use this? So that's mussels. A mussel sits down on a rock in the ocean against tremendous buffeting of wave action and has to stick there. And it does this by putting out these byssal threads all around to hold it in place. So when the mussel is going to make a byssal thread, it puts its foot out and it's sampling the surface. It lays down a plaque, and the plaque has a striated layers of different proteins that are there that have different properties. So my research group does not work on the muscle foot proteins, but Herb Waite's group does, who's in biology here. And what he has shown is that these muscle MFP, original name muscle foot protein, are the MFP5 and 3 are the ones that interact then with the rock. And these are often aluminosilicate rocks, so a lot like mica. So in the lab, we can look at adhesion to mica because we can buy controlled samples of mica. There's up to 30 mole% of tyrosine as an amino acid, and that tyrosine gets hydroxylated to form dopa. So I'll show you that in a second. And the lysine and the tyrosine are often right next to each other. And so here we go. This is what dopa is. It's a catechol like we saw in the microbial siderophores. It's a slightly different kind of catechol. It's what we call a 3,4. So 1, 2, 3, and 4 carbons are hydroxylated as opposed to the 2 and 3 carbons. And it's next to this lysine. So it had been known for a long time that the catechol was essential for the adhesion of these muscle foot proteins to rocks. But it wasn't really understood how this cationic amine was important. And it's hard to make a mutation in a protein and mutate out all of the lysines and expect to see the same type of protein again. But in a small molecule, we could investigate this. And so now we're coming back to the siderophores. So we wanted to know what is the force of adhesion of these siderophore kinds of compounds. And we worked with a group in chemical engineering it was Jacob Israeli's group. And he developed this surface forces apparatus to work in water. And so if you take 2 smooth cylinders of mica and you orient them perpendicular to each other, they contact at a point. And if we coat one surface or even coat both surfaces with a compound of interest, and they're pretty far apart, and we begin to bring them in close together in the open circles, at the point that a compound on one cylinder feels an attractive force to the mica on the other cylinder, there's a jump in. And we can continue to compress to a certain force, and we can let it incubate or wait for a period of time and then pull apart. And the point that the applied force is higher than the adhesive force, then we see a pop-off of the molecule. So this is the adhesive, the force of adhesion. And that's what we're interested in. What is the force of adhesion? Okay, so you can see then here's our molecule, cyclic trichrysobacter, the siderophore. Here's the 2,3-dihydroxycatechol versus the 3,4-dihydroxycatechol of dopa, and they each have lysine. So Herb Waite and Jacob Israeli had done a lot of work on muscle foot proteins. We had a benchmark of what was good adhesion. And if we look at our molecule here, this is in the surface forces apparatus. We bring it together, and we can either wait for 2 or 10 or 30 minutes. The force of adhesion is good. It's around 40 millinewtons per meter compared to the proteins, which are up around 60 or 70. But the weak link is this ester. So for those of you in chemistry labs, you'll know that the ester link can hydrolyze. And if we hydrolyze this whole thing, then we don't have a molecule that could bridge the 2 mica cylinders together. So a student of mine said, let's just synthesize these compounds, and we'll put in a tren and amine core. We'll still keep the lysine, and we'll keep the catechol. And we can make all permutations of this molecule that we want. And so when we ran this molecule in the SFA, we got a force of adhesion that was 90 millinewtons per meter. So that's much even better than the muscle foot proteins. And there's a lot of information on this slide. So I have to tell you, mica is anionic, and it in water or in salt water, it develops a hydration layer, so hydrated potassium ions or cations on the surface. And we can get a measure of that thickness when we look in the open circles of just buffer and we compress it, and we get what's called a hard wall thickness of about 14 angstroms. And you pull apart, and of course the buffer's not sticky, so there's no adhesion, so we see no force of adhesion. But in our molecule, we can see that it gets in beneath this hydration layer, and the hard wall thickness is around 8 or 9 angstroms. And so it's been able to penetrate beneath this in order to stick to the surface. So this is a perfect example then. If we make permutations there can we tease out what's the relative importance of the catechol versus the amine. So Greg Mayer in my group said, okay, here's the original molecule we started with lysine and the catechol. Let's just shorten that. So we'll shorten it by 2 methylene groups. Or let's keep the lysine but not have the catechol, so we have a phenyl group or a benzyl group. Or let's take the amine group away totally, or we could acetylate it and therefore it can't be positively charged. So when we have the molecule with catechol and amine, then there's a good force of adhesion and our smallest hard wall thickness. If we just have the amine, there is some adhesion, not as much, but there's still a narrow hard wall thickness. And if we have catechol but no cationic amine, it's no different than buffer. It can't get beneath that hydration layer to stick, and the hard wall thickness is thick, just like buffer. And so our model is that the amine is important for priming the surface. It's sweeping these hydrated cations away, preparing the surface for the catechol to attach. So you know that a Band-Aid doesn't work in water, and there's a huge, huge need to develop wet adhesive compounds, both for medical applications, but also other kinds of applications also. And so this is a huge project in many different labs at UCSB as well as around the world. So what I've told you about are 2 stories of 2 different metals in the ocean. One, the abundance of vanadium. We see a lot of vanadium in this vanadium bromoperoxidase enzyme, and it's essential for these organisms to survive and thrive, to keep organisms from colonizing the surface of seaweeds. And I showed you this unusual suites of these amphiphilic siderophores that also happen to be photoreactive, and if they have the right kind of group there, produced by oceanic marine bacteria. And then how these kinds of siderophores were really useful in teasing out how the muscle foot proteins are essential for, and the components in these muscle foot proteins that are essential for binding to the surface of mica or other rocks, aluminosilicate rocks in the ocean. And finally, I really have to thank my students. This is a recent photo of my current research group, and then all of the students that came before them that really made these amazing discoveries that was essential for my group to continue working, current group to continue working. So on the vanadium enzymes, on the siderophores, on the wet adhesion. And this has been a fabulous collaboration with Professor Jacob Israelishvili, who recently died, and Professor Herb Waite. So I'd like to thank you also.